Okay, so good evening, participants. Hope I'm audible. Am I audible? Okay, thank you. Good evening. Thank you for joining in for this live practice session for the Applied Environmental Microbiology course. I am Guru Priya, a Prime Minister Research Fellow in the Department of Civil Engineering, IIT Madras. Um, Similar to the previous weeks, we will just practice some questions. So, for that, the course content of the week 6 it had three different parts like symbiosis, uh, about virus, and about applications or the applied environmental microbiology. So in today's session we'll mostly be focusing on the symbiosis and virus part, because with this our basics or the basic content about microbiology is getting over. Um, so like a few part like acid mine drainage bioremediation was bit discussed in the last lecture in the week 6 course content. More detailed explanation and the MCQs discussion or the practice, we'll do it in the week 7 course content. So in here today we'll mostly focus on learning about symbiosis and about virus more specifically. um, Before that, do you people have any specific doubts or questions in the week six or any other previous week's course content? Any small topic or small doubt, anything is fine. If you have any, you can mention. If you don't have, also you can tell it's clear so that I will post the questions and we'll get started with today's session. Okay, I think there is one more participant with us, like Bigna. It was right. Do you have any doubts from any of the week 1 to 6 course content or anything?
All the questions are true or false, ma'am. In final exam, it will contain only true or false, or some any questions it will be there, ma'am.
I guess
Like fill in the
any
blank.
questions can be there.
Ma'am, in the assignment, week questions, week assignments are all are true or false only, ma'am.
Okay. I'll check with this and let you know
Final,
in the next session. Maybe will that be fine? Because I am not responsible for taking up the question paper and setting up the examination. My TA-ship will be about only discussion part. So maybe I can get to know this and revert back to you.
okay, ma'am. Means in our college they are asking only true or false is there. If we, uh, okay, ma'am, we'll discuss in week 7.
Sure.
Okay. Okay.
Okay. So the first question for today, we will start with symbiosis. A very easy question. You just have to mention the two organisms which makes up the lichen. You can just post the answers in the chat box in case you are new to this live session. You can just read the question, post your answers in the chat box. I'll give you a couple of minutes time, and after that we'll just discuss the answer. So this is how the session will go. Right. So lichen, it's a symbiotic association between fungus and phototrophic algae. Cyanobacteria is also a blue-green algae. So fungus and algae or cyanobacteria makes the symbiotic associations. What is an algae? Is algae only unicellular or multicellular? What's your opinion about algae as far as you have heard or read? Okay. Yeah, it is a phototrophic organism mostly. Algae are mostly colored, so they just uh, take up light as their energy source. And mostly the main use of algae in our environmental engineering field is to like it has the ability to feed on carbon dioxide, so it can be used to sequester carbon dioxide from the atmosphere so that we reduce the pollution. One extra information I would like to give is mostly we think because algae comes under this microbiology kind of study, algae can be microscopic or macroscopic, first point, and it can be multicellular or unicellular. Okay, because we tend to have a common misconception about all these things with what we hear in a general notion. So there are algae which can be um, multicellular also and macroscopic also. And fungus, you all know, like fungus has a very large range of species in it. So in terms of microbiology, we'll be learning about microbial fungi. Okay, so yeah, with that I'll go to the next question. This question is basically what the algal partner receives and what the fungal partner receives. The answers I post may be termed differently. You can tell it in your own way. That's not an issue. It's just about the understanding of the concept. Support and nutrition. Fine. Any more answers? Nutrition is fine. Yeah, perfect, because they mostly algae tends to prepare its own food, store it, so the fungi can use it. Uh, the support, that term can be refined a bit. Any other answers from anyone else? Right, substratum and nutrition. Right. Anyone else has any other answers? Okay, so we'll just see. Okay. So the algal partner receives water and nutrients, and the fungal partner receives food or nutrition. Okay, there is a classic difference between nutrients and nutrition. Like nutrition is what we feed on. Basically, it can be a elemental nutrient also, or it can be a complex food kind of a thing. So it depends on the nature of the organism. So the algal partner needs water and the basic nutrients or the building blocks to make the food. Okay, that is received by the algal partner, and the fungal partner is receiving the food, or as Divya mentioned, the nutrition from the uh, fungal partner. Okay, so this is a symbiotic relationship. Why do we call it a symbiotic relationship? What is symbiosis? Both are benefited. Any other answers? Mutualism is a kind of symbiosis, right? So mutually, both the organisms will get benefited, and that relationship is known as symbiotic relationship. Have you heard about synergistic relationship? If you have heard about synergistic relationship, if anyone has heard, can you tell what it is? Because these terms, in terms of environmental science, environmental engineering, whatever, you will come across in one or the other point. So you have to be clear of this. In general terms, also you can tell what is synergism that will exactly apply to this concept. Okay, I guess you don't have an idea. So let me tell you. In symbiosis, what happens is there is a relationship, and we just define that they are benefited. But in the case of synergism, it's kind of an additive effect. It's not addition, it's additive effect. So two organisms interact, both are benefited, and the net benefit that is gotten is more than the sum of the individual benefits. Okay. uh, for example, in the case of a synergistic relationship, if we are going to treat some pollution, okay, so I am putting two microorganisms to treat some kind of a pollutant in water medium, let's take. Okay, so in that, if I use one organism, maybe the rate of the reaction will be uh, some fold. When I use the other organism individually, it will be some number. When I use both of this together, if the net rate is greater than the sum of the individual, then that is known as synergistic effect. Okay. um, and there is one more known as a syntrophic effect. So syntrophic effect is nothing but it's just they help in a sequential manner. For example, one organism produces some product, like I put two organisms in a water containing some nutrients. Okay, one organism is able to produce is able to consume the nutrients that is already available in the water and produce a product. The other organism that is present there will consume the product that is produced by the first organism, and it will produce another product which is again useful to us. Okay, or it helps in the treatment of that water that I am putting in, like something in that context. But there is a sequential utilization. The substrate is utilized by organism 1 and the product of organism 1 is utilized by organism 2. So this relationship is known as syntrophic relationship. These terms generally sound similar, so we tend to get confused with these terms. So that's why I'm explaining it. It's just symbiosis, uh, synergism, and syntrophism. Okay. So any doubts in this? Is was that clear? Was it clear, or you want me to repeat it? You can just post in the chat box. One second. You can just post in the chat box or give a thumbs up if we can move to the third question. Okay, sure. So in this question, basically, we have a stratified lake. So in that stratified lake, the depth varies, and in turn, the nutrient availability also varies. So because of this, a phenomenon can be found in a stratified lake, which was discussed in the lecture. So I express just one term that defines the whole thing. Or maybe if you're not aware of the term, that's perfectly fine. You can just give me a context of that based on your understanding. I'm just asking about how or what are the kinds of microbes that will be present. All these are just general questions in order to check your understanding about the concept. Okay, so if you are understanding it, it's pretty good. If you are not, then I'm here to explain it. So you can just give me answers. It doesn't need to be right or something. So when you give me some answers, I'll understand what you have taken up regarding that topic, and based on that, I'll explain it. Any answer would do. Like, what do you understand from this question? Like, is the question clear? At least you can let me know that in chat box. If the question is not clear, you can just put not clear in the chat box so that maybe I'll try to explain the question better. Okay, assuming that the question is not clear, let me just rephrase the question once more. See, we have a stratified lake. Okay, stratified in the sense it has different zones. So, with varying depth, the area of nutrient availability is also varying. Maybe you can imagine a stratified lake, right? It was explained in maybe 2 3 lectures in the same course. So, in that stratified lake, how will the microorganisms be? Will same microorganisms be present, or will different microorganisms be present? What's your idea about that? If you feel the microorganisms will be uniform, you can just give uniform, or you can give non-uniform. I'll give you options now. I'm audible, right? Do you think same organisms will be present or different organisms will be present in the over the stratification? I guess there are hardly 4 people here today, so you can try to answer. It's not an issue. We are just few people here in the meet. Okay, do you want me to post the answer for this? You can at least reply for this in the chat box. Yes, no, something in the chat box. Can I see? Right. Okay. So, before telling the answer, please let me know one thing. Are you all aware of all these terms and stuff? Because are you all, I don't know your backgrounds. I guess Alageshwari, you are from nutrition science. I remember you telling this in the last session. Uh, so, apart from this, you people can tell me if you belong to varied backgrounds. I'll try to explain it that way, because if I'm going to ask the question, wait for few minutes, no one will reply, and I'll tell the answer, neither you are benefited nor me, right? So I should be giving you something useful in these 2 hours. So please let me know what's your background, or how we should proceed with this. Are you able to understand the questions? Give me a yes or no in the chat box, please. Maybe 2 more people at least respond. Like, are you able to even understand the questions? If it's no, it's completely fine, because people from different backgrounds cannot answer all questions. I'll at least try to explain. If you are knowing this, I'll explain it in a different way. If you're not knowing this, I'll explain it from the beginning. So you can just tell me. Abhigna, I guess that yes, you replied was for that you um, want the answer for this question, but just let me know if you're able to understand the question, or you can also add what branch or what you belong to, what are you currently doing. Okay, my voice is breaking. Maybe some network issue. I'll try to. Oh, you're from biotechnology. So, did you understand this question? What do you think will be the answer for this question? Okay. Okay, that's fine. We'll just move on with the discussion. We'll just discuss few more questions so that you get used to it. So, enables the presence of consortium. Basically, what I was trying to tell was at variable depth, the nutrient availability will be different, and because of that, there will be different microorganisms present. Okay, so these different microorganisms together present in one environment is known as consortium. Maybe I'll explain to you with assuming that you are not aware of a stratified lake or something. See, in a kind of a stratified lake, if you take this as the upper surface that is in connection with air, you will also have few layers below. Okay, sorry for some kind of a worse drawing, but still bear with me. So here what you have is, here you have the air environment. So there is good oxygen transfer here, and in the surface you will have a better aerobic organisms. Whereas here the oxygen concentration is going to decrease further, and finally you will have an anoxic zones and anaerobic zones. Okay, so in anoxic zones, either there will be presence of sulfate, nitrate. So sulfate consuming bacteria and nitrogen consuming bacteria will come into the picture. They will use sulfate and nitrogen and then get energy. In the anaerobic zone, it's again the anaerobic organisms will be surviving. So this kind of a stratification and nutrient availability uh, variations across this from top to bottom is the concept of stratified lake, and this enables the presence of different microorganisms in different layers. Okay, all these together are called a consortium. When you have only a single microorganism, for example, if you are from a microbiology or a biotechnology background, you will know that you will culture E. coli for some purpose, or you will culture Zymomonas for some purpose, Saccharomyces for some purpose. When you are going to do in such a way, that is called a pure culture. Okay, a pure culture is something where only you are going to cultivate a single species of a microorganism. But in a consortium, there are different microorganisms coexisting together. Is it clear about consortium? Okay, with that we will also discuss about lichens. So as you told, lichens is a symbiotic partnership between fungi and algae. So the dominant partner is mostly fungi uh, because it gives fungi majority of its characteristics, from its thallus shape to its fruiting bodies. Uh, Algae, as discussed, can either be green algae or a blue-green algae or brown algae. Any algae is fine. So this is some an extra information if you have not known about lichen. So basically, lichen has these three different types like crustose, foliose, and fruticose. Like any other classification, it is based on their morphology, characteristics, and different stuff. Those details is not very necessary in the point of view of this course. You can just check it out if you are interested to know more about lichen. And here are a few pictures about lichen. If you haven't searched for it or seen it before, this is crustose, this is foliose, and this is fruticose. So yeah, now I'll just go for the next question. First, tell me what is PFU and what is CFU. Okay, and before that, whatever I was speaking till now, was it breaking or was it fine? Um, Is there any issue with the audio or something? You people can respond to the chat box. Like, is there any issue with the audio? Right. PFU is a platform. Okay, okay, fine. Okay. So you can just tell me what is PFU, like expand PFU, CFU, and then just answer this question. This is a true or false question basically. So, uh, in this session uh, that we are doing today, any question that's just a statement is a true or false question. I have not mentioned it explicitly in all the questions, but yeah, if I'm giving a statement, you have to just tell me it's true or false. Right. PFU is plague forming unit and CFU is colony forming unit. Now you can answer the question. Okay, one person has told it's true. Let's wait for others also. Okay. So as you have told, it's true. Nothing but basically, we are just going to have a plate. As we all know, plate is where in the laboratory we culture these microorganisms. So in the case of virus, since they don't replicate by themselves, what we do is we just give them a support to grow on, and uh, finally in our plate the phages will be grown, and we'll be counting them in the name of plague forming units. But in the case of bacteria, what the individual colonies that grow, that we count. So each single bacteria will give rise to a colony. So that one we have to count so that we get the CFU and PFU. For what do we use CFU and PFU? What are they? Why do we need to count them? Why do we need to know CFU and PFU? Okay, in any bioprocess, you have to know the bacterial load basically. So in that case, you will need this count of CFU and PFU. And in case you are testing the wastewater characteristics, so as far as this environmental microbiology is concerned, we'll take estimating the number of viable bacteria. Exactly, that's the answer. We need to know, for example, I give you a water sample. uh, we just live in a locality, and around from 500 people that live, 100 people are affected by some disease, and we suspect the water quality. What do? We just go and see if there is a presence of uh, some microorganism. Immediately what we do is we also go for counting how much microorganism is present. Not only that, when we do water treatment, basically what we do is at the end there are some characteristics that to which we have to stick on. We need to have some amount of BOD, some amount of COD, above which we should not go. If the value is above that, then the water is not fit for you consumption or usage, depending on standards for different purposes. Okay, so in that case also, after treatment also, you have to once again check the count of bacteria that is present in it, in order to certify that it's safe to drink it. Okay, so the CFU and PFU are methods to count the microorganisms or the viable microorganisms, basically in the case of bacteria, for various purposes, and what I told was one of them. Okay, so with that we'll move on to the next question. This is also a true or false question. Can ruminant animals digest cellulose? Yes or no? One. Okay. Two. Two true. Okay. So yeah, we have two true and a bit of explanation, because cows have specialized structure that allows them to digest cellulose effectively. Uh, Let me also rephrase this in, yeah, rumen. Okay. Let me also rephrase this once again. Can ruminant animals digest cellulose all by themselves? The question was, can ruminant animals digest cellulose all by themselves? Anything, yes or no, just give me a yes or no so that I'll start discussing about this. Very simple. I just want to know what your understanding and perspective is. So, okay, I'll give you three options now. Can ruminant animals digest cellulose all by themselves? Yes, no, maybe. You will have some answer, right? It may be right or wrong. You just give me some answer. It's fine. Alageshwari and Bigna, what do you think? Yeah, yes, ma'am, they can. Okay. Okay, so this is what the confusion is about, and this is why I posted this question. You all know that humans are almost not capable of digesting this, like cellulose and all, because uh, we don't have this cellulase enzyme. The cellulose is a bit difficult for humans to digest, basically. So yes. So ruminant organisms also they can digest cellulose. Yeah, they take cellulose rich food, they digest it, derive energy out of it. That's a different story. But ruminant animals cannot digest cellulose all by themselves. So that is why we are studying that in our microbiology course. Why are we studying about ruminant animals in our microbiology course? Just because, yeah, exactly, ruminal microbes. Just because ruminal microbes are the ones that are helping in the digestion of cellulose. These ruminal microbes are present in the rumen. So what basically happens is cellulose is being ingested by the animal, and it goes into the digestive system, wherein these ruminal microbes are present and they are capable of cellulolysis. Okay, so they have cellulolytic enzymes like cellulase maybe, and then they help in the breakdown of cellulose. If you are going to eliminate the microorganism from the digestive system of that animal, the animal itself cannot digest cellulose. Okay, so in the general context, ruminant animals can digest cellulose, but they are not doing it by themselves or by the enzymes produced by the animal itself. Instead, it is done by the microorganisms that are present in the digestive system of these ruminant animals, and this is the symbiosis, because the microbe gets its substratum food, and in turn the ruminant animal gets the cellulose digested. Okay, this is symbiosis in this case. Is it clear? So the answer is true. Ruminant animals can digest cellulose, but not all by themselves, with the help of ruminal microbes. Is that clear? Any doubts? Okay, fine. I'll pose the next question. Very easy question now. Ruminant animals undergo dash gut fermentation. It's a very easy question if you have seen the lecture videos. Ruminant animals undergo what gut fermentation? It's not rumen. It's just about what's the type of fermentation that happens based on the structure of the gut. So based on the structure of the gut, what kind of a Okay, yeah, Abhigna is right again. Ruminant animals undergo foregut fermentation. So there are two things, right? Foregut and hindgut. So in case of foregut, as the name suggests, in the starting part of the gut, the fermentation happens. In the case of hind, it's the structure is at a bit lower downside of the system. So ruminant animals are generally foregut. Okay, sorry. Here, I think I have displayed the answer also together. Okay, I'll just tell about this. Biomagnification is a prerequisite for bioremediation. No, it is not a prerequisite. So, do you know about biomagnification? What is biomagnification? I think even I have told this in one of the previous sessions. Increase in concentration of a substance. Oh, is that biomagnification or some other phenomena? This is also one of the phenomena, but I'm just confirming if this is biomagnification or something else. Rise or increase the contaminated substance. This has a very peculiar characteristic that defines magnification. Okay. So before discussing about magnification, let me also put on one more question and we'll discuss. What is bioaccumulation? Have you heard about bioaccumulation? Because these two terms are generally used together. They mean different things. Okay. Assuming that I have to explain, let me start. Increase in concentration of a substance generally is bioaccumulation. For example, we have a fish species in a lake. Okay, so the lake is contaminated with, let's say, some heavy metal. Okay, so first what happens is, as the fish is going to take up nutrients from the lake, it will also take up this heavy metal. And let's say this is just a very raw example. Okay, don't worry about the figures and all, they're not exact. So let's say on the first day it accumulates around 1 microgram of that heavy metal, and in the second day again this just keeps increasing. At the end of 20 days, let's say, around 1 mg is being accumulated. 1 mg is being of heavy metal is being accumulated in the body of the fish or organism. So this phenomenon is known as bioaccumulation. Okay, over a period of time, something gets getting accumulated, a pollutant getting accumulated in the body of the organism is known as bioaccumulation. Biomagnification is something related to the ecosystem. Okay, the chain, food chain basically. So let's say a fish is contaminated, a small fish, let's say, it will have very less amount of that pollutant in its body. But let's say a big fish that consumes this small fish will eat around 10-20 of this fish. Then what happens? More amount is being accumulated in the body of the second, that higher fish, or the bigger higher order of fish. And the larger fish is being consumed by some birds, let's say. So that bird will have much more of that accumulated, and humans eating that bird will have much more of the pollutant being accumulated in the body. This kind of increase in concentration over a food chain is called as biomagnification. So there are two things, remember. It's biomagnification and bioaccumulation. Bioaccumulation is just increase in concentration over a time, but in case of biomagnification, it also involves the concept of ecosystem, where of food chain in it with it. Okay, is it clear? Give me a thumbs up if that is clear. Oh, okay, fine. Right. So with that, I'll go to the next question. Hope answer is not that. Yeah, fine. You can try this. Very simple question. You can just give me the answer in the chat. This is again a true or false question. As I mentioned, any question which is a statement is a true or false question in our today's session. Is this true or false? Any answers or any opinions? Okay, two answers there. Okay, so this is a very simple question. One way to treat acid mine drainage is to add a reducing layer to reduce the sulfate. It's just about chemistry. There is no much of microbiology here. Okay, so it's just that we are going to treat the acid mine drainage. So acid mine drainage is usually having a acidic pH and it being drained out. The metals being drained out. So you can add a reducing layer to reduce the sulfate. Sulfate is SO4 2 minus, right? So when they are going to combine with H plus ions, they will again produce H2SO4. So this is the basic cause of this acidity. So we can add a reducing layer to it. The next question. Commensal bacteria are found only in the biofilms. Is it true or false? What are commensal bacteria? Okay, I got two false for this. So for anyone who couldn't understand the commensal bacteria, can someone who knows it put it in the chat box? Okay, so it's false as all of you have told. Commensalism is something where there's a relationship between two organisms where one is benefited and other is neither benefited nor harmed. So one is plus, another is neutral, zero. Okay, so that kind of a relationship is a commensal relationship. Commensalism is also found in human body. Like we have few microorganisms in our body with whom we share this commensalism. Okay, so that is one example. So it's not found only in the biofilms. Very easy question again. Viruses are neither living nor non-living. Is it true or false? Very simple question, right? This is the basics of virology, viruses. It's non-living. Okay. Okay, so we have false, true, false. Answer is true. Viruses are in the gateway. Generally, in the scientific study, what we tell is viruses are in the gateway. They are neither living nor non-living because um, they cannot replicate on their own. But when you put them into a suitable host, they can replicate. Non-living organisms, if when you are going to strictly tell them as non-living, then they will never replicate. Like then whatever you do, they're not going to replicate and multiply themselves and put a copy of them. When it is living, then the properties are different, but this is in the gateway of living organisms and non-living organisms, and they do not fall into either of this category. Okay, so this is very important to know that viruses are classified as neither living nor non-living. They just stand in the gateway between both, because when they have a suitable host, they will multiply, they will act as something, maybe kind of a living thing. When there is no host, they act kind of a non-living. So okay, virus is defined as, this question is in the context of that what element it is. Like in the case of a plasmid of microorganisms, we tell them extra chromosomal DNA, something like that. We define them in that way, right? Similar to this, how can, just an analogy, like similar to that, what is virus called? Right, we have two interesting answers. So it is a subcellular genetic element, yeah, and also equally true that they are obligate intracellular parasites, because obligatory because they require hosts to survive. So that is why. So this is why I give open-end questions. See, if I give four options, then I'm confining you to my way of thinking. But in this, we are getting some different answers where both are equally true. So we get to know something we don't know, or something addition in addition. Okay, so this is a very good point for this question. Virus can be defined in multiple ways. One is it can be called as a subcellular genetic element. Other is it can be called as an obligate intracellular parasite. Right? Since it doesn't have its own machinery and it requires a host for replication, it's known as a subcellular genetic element. A virus essentially has either a RNA genome or a DNA genome and replicates correspondingly. Okay, fine. I'll post the next question now. Right, as all of you have rightly told, they are extracellular virus particles. Okay, they are extracellular virus particles. They don't have a protein code generally. Mostly they have only single stranded RNA. So this is what it is. In with this, I just wanted to know if you have noted down the lectures for this topic. Regarding viral evolution, there were three hypotheses. To be very clear, there are three hypotheses. You just have to name all the three. Very simple. This is just a memory based question. No like scientific concept involved. Just the three hypotheses name related to viral evolution. I'll just wait for a few more minutes so someone can answer. I'll just give wait for a few more minutes. Okay. So as per the lecture, it was alternative hypothesis, reductive evolution, as well as uh, Misel's based evolution. But in general, you will hear about virus first hypothesis, escape hypothesis, and reduction hypothesis. Okay, so I'll repeat. Alternative, reductive, Mises-based evolution is the term used in the lecture, but in general you will hear them as virus first hypothesis or cell first hypothesis. So in the cell first hypothesis, virus first hypothesis means like virus was the first organism that was there even before uh, Luca known. Okay, after that only the cells and all came. So that kind of a hypothesis, the virus first hypothesis. Whereas there is one more called the cell first hypothesis, where there is origin of cells prior to viruses, and this includes escape hypothesis and reduction hypothesis. Okay, so the alternative hypothesis states that the genetic fragments that code for essential cellular functions escape and gain viral genome. These genes replicate in cellular host and form virus-like particles. Okay, so what they are trying to tell is we have genetic fragments. They code for cellular functions, meaning they code for proteins or enzymes that are essential for cellular functions. Coding for a function is coding for the enzyme that makes the function feasible, and they gain the viral genome. These genes can replicate in the cellular host and they will form the virus like particle. So this is the alternate hypothesis. Whereas in the reductive hypothesis, primitive cells with reduced size and genome get encapsulated by endosymbionts. Endosymbionts are nothing but they have a symbiotic relationship and one is present inside another. Okay, so that is endosymbiont. I think you would have heard about endosymbiotic theory where we talk about this chloroplast and mitochondria. Maybe that would be a interesting read if you are interested in kind of evolution and stuff. Uh, Endosymbiosis theory is one. So here what they are telling is primitive cells that had reduced size, the size of the cell as well as the genome, they get encapsulated by endosymbiont and develop into viral-like particles. So this is what is the statement of reductive evolution. Whereas in the micelles-based evolution, they tell that the primitive atmosphere had micelles. Micelles are some small particles. Okay, they trap nucleic acids. Maybe when you heard about oil or something, you would have heard about micelles in that chemistry part. So in that, these nucleic acids are trapped over time, what happened was they could eventually find ribozymes. Ribozymes are enzymes that uh, help this nucleic acid. So they had this ribozyme activity and they formed protovirus. Okay, so these are the three hypotheses that are said to be applicable to viral evolution. Okay, is that clear? Can we go to the next question? Thumbs up, please. Oh, okay, fine. This is again a true or false question. Okay, so I hope you are all aware about mutation. Mutation is a change in the nucleic acid sequence, DNA sequence basically. So when there is a nucleic acid change, eventually there can be a change. The mutation can be addition, deletion, anything. To be very to tell it very simply, it can be addition, deletion, and there are many more. So when it's going to be addition or deletion, the frames can shift, and because of the reading frames can shift, and because of that the transcription and translation will not happen as expected for that gene. They can produce a different protein, so the functionality may change, and that could lead to a very devastating effect. Okay, so basically, mutation is a change in the DNA sequence. Mutation rate is how fast this mutation occurs with respect to time. So, rate we generally see how much mutation occurs with time. Here we have it is mentioned that the mutation rate directly proportional to genome size. Genome size is a number of base pairs that is present. So usually we denote the DNA size as this much base pair. Like for example, in human we say it's 3.2 billion base pairs. So that many base pairs are present in the human DNA or human genome. Okay. So mutation rate is directly proportional to genome size. As they have rightly answered, it is false. Mutation rate is inversely proportional to the genome size. So when the genome size is very fast, the ability to manipulate it or it getting mutated is higher. So the mutation rate is high when the genome size is low and vice versa. Okay. I hope we are clear with what is a genome size, what is a mutation rate, and the fact that the mutation rate is inversely proportional to the genome size, right? Okay, with that, I'll post the next question. Okay, viruses with capsids and nucleic acid only are called naked viruses, and they can also be termed as nucleocapsid. As mentioned, as this open-ended question, you can have multiple right answers. So just they just have nucleic acid and capsid, so it's like a nucleocapsid or a naked virus. When there is one more envelope present outside, we call them as enveloped virus. As mentioned, so today's session will mostly be on your memory and understanding to a little extent. Nothing much of experimental or theory in it or an underlying principle in it. Okay. So with that, we'll go to the next question. I'm sorry for the typing mistake. It's viruses that can switch from single-stranded RNA to double-stranded DNA R dash. Strongly mentioned as RNS. It should have been RNA. Any more answers? We have retroviruses and hepatnaviruses. Okay, so yeah, we now have majority retroviruses. The answer is retroviruses. To be very clear, RNA viruses that contain single-stranded RNA that can convert it into DNA. That is, they generally have only RNA, but they have enzymes that can convert it into DNA. This kind of viruses are known as retroviruses. The other way around, predominantly it is DNA, but they have this capability to convert the DNA into RNA. That kind of viruses are known as hepatnaviruses. So just be careful. In the case of retroviruses, they usually possess RNA but have the ability to convert this RNA into DNA. Whereas in the case of hepatnaviruses, they usually have DNA, but they tend to uh, transcribe maybe into RNA. Okay. So you have to be careful about what is predominant in hepadna and retro. Just remember one, other is just the opposite of it. RNA to DNA, it's retroviruses. Okay. So since you have answered it in the previous question itself, viruses that can switch. I'm sorry. I think again this is a typo. As I told, it's from DNA to RNA. It is hepadna viruses. Okay. So here, yeah, dash is the most essential requirement of in vitro virus growth. I hope you are clear with the term in vitro. Anyone hearing this for the first time or do not know what in vitro means? Okay, so any of you for whom you are hearing it for the first time, in vitro means in a culture plate or in a glass vessel. Okay, glass vessel or a medium, like kind of a petri dish, test tube. In the case of bacteria, if you are going to grow them in a plating technique, is a in vitro technique because we're trying to grow the bacteria in a glass dish. Okay, we have two answers, cell line, host. Okay. Anyone else would want to try? Media. Yeah, it is an interesting answer, but we can discuss about it. Okay, assuming that there are no more answers for this, you are right. We just need cell lines or host cells. You're absolutely right. Media, yeah, it is a requirement, but the difference here is in the case of bacteria, media is the main requirement. Like all the nutrients, the carbon source, the nitrogen source, whatever the microbe is going to uptake is going to be from the media. So media is a very critical, very essential requirement. But in our case of this in vitro virus growth, what happens is we will first need the cell lines or host cells in which the virus can replicate. If you are not going to have this cell lines or host cells, the virus is not going to replicate. Okay, so that's one important point. And one more thing, even if you are going to provide the media, virus cannot take it up directly and grow. Okay, the media is not required by the virus, but instead it is essentially required by the cell line or the host that is going to help the virus to replicate. Virus has nothing to do with the media. It's not going to uptake, it's not going to metabolize or do anything. It's just that virus requires the cell lines, and the cell lines or the host cells in turn require the medium. They need glucose or any carbon source, energy source for them to consume, to have their housekeeping genes working, to have their metabolism normal, so that they can perform the function of protein synthesis, basically the central dogma. So in that case, they also tend to take up the viral genome, and then they replicate the viral genome. They can also translate the genome to produce this capsid and all, and finally they assemble, and then um, viral particles, many viral particles are released. So the catch here is virus requires cell or hosts only to replicate, but the cell or host cells require media to replicate. Virus has no direct connection with the media. So here is the next question. It's a true or false question. Very simple if you understand the curve. Like we had the bacterial growth curve, you have the viral growth in mind, you can answer this question easily. We have two answers, one true, one false. So if I get one more answer, I'll just discuss the answer. We'll just see which side is favored, false or true. Anyone more having any opinion about this? It's true or false? Okay, so we have two false and one true, and obviously the majority is right. It's false. If you are going to see the curve, it is going to have um, a latent period where virus is added and there is no increase in the viral particles or the PFU. Basically, what you plot is going to be PFU versus time in here. In bacteria, it'll be CFU versus time, right? So here what we are going to plot is the plaque forming units versus the time. So in the latent period, there will be no increase in the viral number. It's just going to stay constant, a straight line, and then it's going to increase. Okay, why is this latent period happening is it's just a time for the host cell to get its machinery ready. The host cell has to first the DNA has to integrate. If it's going to integrate in the host genome, it has to integrate, and then there is some time that is taken for the metabolism of that organism, and then only it starts replicating, right? So it starts replicating, transcribing, and translating. So these process take some time. During that time, the virus just stays inside, and after this, um, once this transcription and translation starts, then okay, more of this viral genome is multiplied, viral protein codes are produced, and they can be packed for the next set. So it's just in the latent period the virus enters. So when it enters, the genome has to enter, or when the virus itself enters, the it has to lose its protein code. All these mechanisms take some time, so that period is known as the latent period. And after that, once the machinery is ready, the viral genome also replicates, and the viral proteins are produced, and virus is again packed. So in this period, we call it as the assembly and release period, wherein the viral number uh, increases. So this is the next question. This concept you have studied in week 5 also: conjugation, transformation, transduction, when you studied about gene transfer. Very simple question. I'll just wait for 2 more answers. Right. So it is phage. We call them phage. In transduction, the genes are transferred from a donor bacteria to a recipient bacteria. So the virus that infect bacteria are called as bacteriophages. So here we term it as phage. It's just to introduce you to all terms related to this concept. They may mean one and the same, but still you have to know all the terms. And in some cases, there will be intricate differences between the statements where only few situations are right to use a particular term or a statement, and you can't use it in others. In that sense, you have to be clear. So you have to basically be clear about the concept so that you don't go wrong. Okay. This is again a very simple question about viral replication. Viral replication can be either, we just quickly go through next. I think I have 4 questions. So, right, lytic and lysogenic. So, in the case of lytic cycle, what happens is, uh, again, is the phage infects, uh, and its genome is released, it gets replicated, the viral proteins are produced, and they are packed, and finally the host cell is broken down, okay, for the virus particle to be released. Whereas in the case of lysogenic cycle, the host is not, there is no like physical damage or something to the host in that case. Okay, so that is the lysogenic cycle. The key enzyme that is present in the retroviruses, kind of discussed regarding this when we discussed about PCR. I think that was in week 4 or 5. I don't remember. Yeah. Great. Reverse transcriptase, integrase, protease, right? But generally, when you are going to remember about retrovirus, you are going to remember about reverse transcriptase that helps. That is reverse of transcription. This enzyme helps in a process that is reverse of transcription. What is transcription? It's DNA to RNA. But when you convert RNA to DNA, the process is known as reverse transcription, and the enzyme that helps is known as reverse transcriptase. As I told, this is just an extra information if you want to explore more. Um, no, they don't have any polymerase or anything as such. Polymerase for that and all, they'll depend on the host cell only. They just can convert their RNA to DNA and DNA to RNA, something like that. So on that context, they will just have reverse transcriptase. Retroviruses, basically what they have, single-stranded RNA. Sorry, they have to convert their single-stranded RNA to double-stranded DNA. So for that purpose, they use this reverse transcriptase. And we also have a dedicated PCR type known as RT-PCR, which is the reverse transcription PCR. It is it depends on where you want to apply. Maybe in the library construction, genome sequencing, and all. Reverse transcription can also be used in some cases. So if you want, you can just read about it. I just wanted to introduce that to you. Right. So that's the answer. Tumor. Polyoma viruses can be of different types. They can cause different things and different diseases. But as far as our course and lecture was concerned, it is very important to note that polyoma viruses are able to cause tumor. I hope you are aware about what tumor is. This being a civil engineering course, I'll just tell you, it is uncontrolled like cell division. Like it's not cancer. Cancer is exactly uncontrolled cell division where your cells keep on multiplying, but tumor is something before that. It A mass of cell grows and then the growth stops and something like a lump of cells grows. So that is a tumor. Polyomaviruses are capable of producing this tumor. This is again a true or false question. Antiprotozoan drugs can, it's not attach, it's attack human cells also, or attach anything, any ways you can take it. So we get some diseases from protozoans also, right? So if we take the drugs for it, will it affect our cells also? It's mentioned that the drugs that we take for protozoan diseases affects our cells also. You just have to tell if it's true or false. False. Okay. True. We'll wait for one more then. Anyone else has an opinion about if it's false or true so that we can see what's majority here? Anyone else would like to try? Okay, so if it's going to see, we are infected with protozoan disease, we are taking the drugs, and why will it affect us? If it's true. Any answers for that? Okay, two people are telling it true. But yeah, humans are affected by the disease. Only to cure out of the disease, we will take the drugs. Why will it affect the human cell itself? Is there any rationale behind why it does so? Very simple one word answer can do. I'll just explain it. On what basis you have told it's true? I'm just asking like what on what basis you are uh, uh, thinking this statement to be true. Okay, so this is true, and the rationale behind me telling this is because protozoans are eukaryotic mostly. Okay, so protozoans are eukaryotic. So human cells are also eukaryotic. So when you are going, when you want to give an antimicrobial drug, what you are basically going to do is target any of its structure or metabolic function such that it doesn't work or it disintegrates, right? So in the case of anti-protozoan drugs, they are going to hinder the protozoan functioning or the structure or the function. It being a eukaryote and humans also being eukaryote, this can sometimes mismatch. It could potentially mismatch, right? Logically. So that is why anti-protozoan drugs can attack human cells also. Okay. So this is the last question for the day. So you can try answering this and we'll wind up in few minutes. Again, a true or false question. this question is based on understanding and logics. That's it. So you just have to think what is in enteric protozoa and what are vectors, and will both of this go hand in hand. My God, all of you are telling it's true. Any more answers? I'll just wait for one more minute. Okay, enteric organisms. What are they? They affect the intestine, right? We have studied about enteric microbes like Salmonella typhi, it causes typhoid. So amoeba, amoebiasis, amoeba is a protozoa again, right? So all these are enteric. Okay, vectors means something that transfers these microorganisms. Okay, so in the case of enteric, mostly what happens is we have a contaminated food or a contaminated water, and we are just going to ingest it. So when we are going to ingest, what happens is it directly enters our system, colonizes our gut, causes the disease, and we fall sick. Whereas there are a few protozoans like maybe plasmodium. You have heard plasmodium falciparum, which causes this malaria and all. Like they require vectors like mosquito. Okay, so what happens is they stay in the gut of the mosquito, and when the mosquito bites us, it just injects the pathogen into our body, which then like replicates and hinders our system. Okay, so when the microorganism is not directly entering but entering through some other, with the help of other organism or other helpers, those helpers are known as vectors, and that is vector based transfer. Whereas mostly in the case of enteric, what happens is we are directly going to ingest it through our food or water, so they will not require vectors, and this statement is false. Is that clear? Okay, so with that, I'll just we are just coming to the conclusion of this session. I had 25 questions and we had discussed all of it. Thank you for your active participation in this discussion session. So I'll just wait for a few more minutes to see if someone has any doubts, any questions, or any clarifications from all the 6 weeks, or particular to this session, or some doubts about the next session. Any doubts or suggestions about the live sessions can be given either in the chat box or can be told by unmuting the mic. Okay, I guess you don't have any doubts. So can you just give me a thumbs up quickly if you don't have any doubts so that I can close the session, right? Others, are you having any doubts? I just got a thumbs up. Okay, so with that, we'll close the session for today, and we'll meet on next Tuesday. Thank you for joining in. Thank you for your time and your attention and the interaction. Wishing you a great week ahead. Thank you all for joining.